All right, this is lecture 2 for microbiology. This one is a review of chemistry. Most of this stuff you have probably seen before, so don't be too concerned if you're looking at it and you feel like it's really easy and not anything new. It's just a good review of chemistry here. This lecture is going to correspond to Bauman's text chapter 2 as well as the Tutora text chapter 2. Matter is anything that takes up space and has mass, and chemistry is just the study of matter. Atoms are your smallest unit of chemical matter. When we look at the atomic structure, the easiest thing for us to use in biology is the Niels Bohr model that was come up with in 1913. It's a simple model of the atom with the nucleus in the center and then the electrons in shells around the outside. It's sometimes called the planetary model. The electrons are negatively charged subatomic particles that are going to be located in these shells around the outside. This first shell likes to have 2, the second shell likes to have 8, and the third shell likes to have 8. When you go looking at atoms beyond that size, this model doesn't work as well, so we typically only look at the smaller atoms with this model. Your protons are positively charged and they're located in the nucleus, as well as the neutrons, which are uncharged or neutral particles in the nucleus. Your element is going to be matter composed of a single type of atom. When you look at the periodic table, the symbol, which is represented by letters in the box, is going to represent the element. Your atomic number is going to tell you the number of protons in the nucleus. Your atomic mass is the sum of the mass of the protons, neutrons, and electrons. To get this number, you add the protons and the neutrons. The electrons are very small, so they're not considered a significant contributor or added in there. This is also called the atomic weight, and it's in AMUs or atomic mass units. These units are also known as Daltons. Your electron shells are going to be the regions that correspond to the different energy levels. Isotopes are going to be atoms of a given element with a different number of neutrons in the nuclei. So they will have similar behavior, but they will have a different mass. Your electron configurations are the arrangements of the electrons. This is what determines the chemical behavior. Of particular importance is the valence, or the electrons in the outermost shell. These are the ones that are going to interact with other atoms and will give you its combining capacity. When we look at organic versus inorganic in chemistry, organic is the chemistry of carbon and hydrogen. Your inorganic molecules will include all of the elements. Carbon is involved, it's just not the star of the show like it is in organic chemistry. A molecule is going to be 2 or more atoms of the same element, where a compound is 2 or more atoms with different elements. The chemical bonds are the attractive forces that are going to combine the atoms or hold them together. The first one we look at is the covalent bond, which involves sharing a pair of electrons by 2 atoms. With the covalent bonds, you can have polar or non-polar covalent bonds. Polar covalent bonds are going to have atoms with different electronegativities, so the electrons will spend more time around one atom than the other. In a non-polar covalent, the electrons are going to spend equal time around each nucleus. Electronegativity is just the attraction for electrons. Here, when we look at this picture, this is going to show an example of a covalent bond. So these 2 electrons here, their shells are going to overlap slightly here, so that each one will have those 2 electrons be within its shell a shared amount of time, and that's going to be what holds them together. So the electrons are represented in red. The other type of bond we have is the ionic bond, which involves the attraction of positive and negative charged ions. The cation has the positive charge, the anion has the negative charge. Salts are the crystalline form of the ionic compounds, and when we dissolve these in water, they're electrolytes. Here you just have the dissociated ions surrounded in water. This is an example of an ionic bond. So here, one of the atoms will lose an electron to take on a +1 charge. One of them will gain an electron to take on a -1 charge. So then you'll have a positive and a negative charge be pulled together. Hydrogen bonds are going to be the result of the attraction of partial positive and negative charges. Here with water you can see where you've got the oxygen here sharing with two hydrogens. The electrons that are shared between the hydrogens are going to spend a greater amount of time closer to oxygen, leaving the hydrogen with a relatively positive charge, a partial charge on the ends. Because the extra electrons are going to be closer to the oxygen, you have a relative partial negative charge on the end of the oxygen. The mole is the molecular weight of a substance expressed in grams. It's 6.02 times 10 to the 23rd particles of something. When you look on the periodic table, your molar mass is going to tell you the mass of one atom in AMUs, or it will tell you the mass of 1 mole of the substance in grams. 1 mole is going to be 6.02 times 10 to the 23rd particles of that substance. In chemical reactions, you have reactants converted into products. It's going to involve making and breaking chemical bonds. Your reactants are the atoms or ions or molecules at the beginning, and the products are the atoms, ions, or molecules at the end. We have different types of reactions. There are many more types of reactions than what we will cover in here, but these are ones common to biology. With a synthesis reaction, these are going to form larger, more complex molecules. One example is a dehydration synthesis. Here, two molecules covalently join together and they will produce water. The water molecule is removed from there, which is why it is called dehydration. An endothermic or endergonic reaction is going to be one that requires energy or absorbs energy. Anabolism is going to be synthesis reactions in an organism. Decomposition is going to break larger molecules into smaller ones. A reaction that is considered exothermic or exergonic is going to release heat or energy. One example is a hydrolysis reaction. Here the covalent bond is broken and the ionic components of water are added to the product. So the hydrogen ion and the hydroxide ion are added to the product. Water is put in to break the molecule apart. Catabolism is going to be just decomposition in an organism. With exchange reactions, you're going to transfer atoms. They're going to be moved from one molecule to another. An example would be molecule A plus molecule BC will form molecule AB plus C. All the components are still there. They're just combined together in a different order. Water is important in any living organisms. It makes up 50 to 99% of most organisms. It has two polar covalent bonds. These polar covalent bonds allow it to do hydrogen bonding, and this is going to account for a lot of the properties of water. Water has a cohesive surface for tension. It's a good solvent. It absorbs heat. It has a wide temperature range as a liquid, and it also participates in reactions. One type of reaction you see with them is acids and bases reactions. Acids dissociate into one or more hydrogen ions and one or more anions. Examples would be hydrochloric acid and sulfuric acid. Bases are going to be molecules that bind with hydroxide ions when they're dissolved in water. So the hydrogen plus the hydroxide would form water. There are other definitions for acids and bases in chemistry. We're just trying to keep it nice and simple in our overview here. pH is the potential hydrogen or percent hydrogen. It's on a logarithmic scale, so if you have one whole number increase, it's going to be a 10-fold increase. So when we look at the pH scale here to the right, something that is a pH 4 is 10 times more acidic than a pH 5. A pH 3 is 10 times more acidic than a pH 4, 100 times more acidic than a pH 5. And it's going to be the same thing when you go the other direction. pH 11 is 10 times more acidic than or 10 times more basic than pH 10. pH 12 is 100 times more basic than pH 10, 10 times more basic than pH 11. Salts are going to be compounds that are going to dissociate in water. Buffers are an important substance in biology. They're substances that prevent a drastic change in pH. They're able to help absorb part of that change. Here's a link that I recommend watching showing an animation on buffers. So again, any of these links in the notes, I do recommend you follow them and watch the animations. I don't include them in the lecture videos because having these on YouTube, it comes up as a copyright issue if I link to other websites. The organic macromolecules, these are very large. They're going to include lipids, carbohydrates, proteins, and your nucleic acids. The functional groups are going to be certain atoms that appear commonly in groups. There are several of them in chemistry. We really only look at a few in biology. The monomer is going to be the simpler units that make up these larger organic macromolecules. So we have subunits of proteins, carbohydrates, and nucleic acids. The polymer is going to be the chains of monomers joined together. If you think of the monomers like being individual Lego pieces, the polymer is the structure that you build with the Legos. Lipids are the first example that we use here, and they are actually a non-monomer. They're almost entirely carbon and hydrogen, so they are hydrophobic. Fats are made from dehydration synthesis. They're going to include your triglycerides. With fats, what we're looking at is the amount of saturation, the number of hydrogens on the carbon bonds. Over here to the right, you can see where this one is saturated. Each carbon is attached to the maximum number of hydrogens. It's not sharing with any neighboring carbon more than once. When you have monounsaturated, you have one unsaturated bond. Polyunsaturated is multiple unsaturated bonds. Down below here you can see an example of something that's unsaturated. You have a double bond between the carbons here. It's sharing electrons twice, and it puts a little kink in the molecule. Your phospholipids, these are two fatty acids that are going to be attached to a glycerol with a phosphate, and they'll have both hydrophilic and hydrophobic ends. This is our example here of a phospholipid. This represents your fatty acid tails and your phosphate head. This is more of the molecular structure. So phospholipids, we see these in cell membranes. Waxes, these are going to be one long chain fatty acid covalently linked to a long chain alcohol by an ester bond. They're completely water insoluble. Certain organisms like Mycobacterium tuberculosis will make waxy walls. So your sterols or steroids, these are going to have 4 rings with 5 to 6 carbons each attached to each other. The most common one in animal cells is cholesterol. Plant cells do have other sterols, they just don't have cholesterol. This is an example of what cholesterol looks like here. Carbohydrates, these are going to be made up of carbon, hydrogen, and oxygen. They're going to be in the proportions of 1 carbon, 2 hydrogen, 1 oxygen. This little n here is going to represent how many carbons you have in there. So you have a triose, which is going to be 3 carbons in there. You would have 3 of everything in the parentheses. So it would be C3H6O3. They're used for energy. They make up the DNA and RNA backbone, as well as a component of cell walls. Monosaccharides are just simple carbohydrates. They're going to be hexoses and pentoses. They typically end with OSE. A disaccharide is going to be 2 monosaccharides linked together. Polysaccharide is going to be several monosaccharides linked together. They can be branched or unbranched. Peptidoglycan is one that is important, that is linked with amino acids in bacteria. Oligosaccharides, oligo means few, these are composed of 2 to 20 monosaccharides. Proteins are going to be composed of carbon, hydrogen, oxygen, nitrogen, and sulfur. They're used to make structures, they're used as enzymatic catalysts, help perform regulatory functions, produce hormones, make up genes, involved with transport, antibodies, complement, and bacteriocins. The amino acid is the protein monomer. Over here we can see an example of a peptide bond where you have 2 amino acids next to each other. You're going to have the hydroxyl group from one and a hydrogen off of the amine group from the other be removed through dehydration to produce water, and it will produce the peptide bond that will join the 2 together. So the peptide bond is just the bond between amino acids. and it is a covalent bond. Stereoisomers, these are going to be mirror images of each other. They will occur in both D and L forms. Amino acids are almost always in the L form. There's a little animation here that shows you chiral molecules. They're stereoisomers because they're going to spin light in opposite directions. When we look at the protein structure, there are different levels to the structure. Proteins are going to be unbranched polypeptides linked in a specific order determined by the genes. Your primary sequence or primary structure is the sequence of amino acids in the protein. Once the primary sequence is established, it will spontaneously fold up into the same shape every single time in the same conditions. Your secondary structure is going to involve ionic and hydrogen bonds. These are going to cause alpha helices and beta pleated sheets to form. Your tertiary structure is your three-dimensional shape. It's not repetitive like the secondary structure. Here, the R groups are going to form your covalent and ionic bonds. You may see things like disulfide bridges. Quaternary bonds, these are going to involve two or more polypeptide chains linked together. You can almost think of these being like an origami project where you are going to fold and then you're going to do additional folds on top of it, and you may even fold up multiple pieces of paper to make your final project. Denaturation is when a protein loses its characteristic shape. This is due to changes in temperature, pH, or salt concentrations. This is an animation of a peptide bond forming here. Our nucleic acids are going to be the DNA and RNA. The shape of DNA is a double helix. It's in the shape of a twisted ladder. The sides or the backbones of the ladder are going to be made up of sugar and phosphate, and then complementary nucleotide pairs are going to make up your rungs of your ladder. Your nucleotides are going to be the monomer of the nucleotide. They consist of a phosphate, a pentose sugar, and a nitrogenous base. The nitrogenous bases would be adenine, thymine, cytosine, guanine, or uracil. The purines are going to be in double rings. They're adenine and guanine. So a quick little way to remember those is pure as gold. These are your examples of purines. The pyrimidines are only going to have a single ring. They're going to include cytosine, uracil, and thymine. DNA is going to form antiparallel strands. They run in opposite directions. One end is going to go 5' to 3', and the other end goes 3' to 5'. The 5' end is specified by carbon number 5 being attached to a phosphate, and the 3' end is specified by carbon number 3 being attached to a hydroxyl molecule. This is a quick little animation here that shows protein structure. When you denature the protein, you disrupt its shape. It can be permanent or temporary. It can be caused by heat, pH change, salt concentration, anything that would interrupt the shape. An example would be hard boiling an egg. So when it's raw, it's going to be slimy, soft. When it's hard boiled, it will turn white and become firm. ATP, this is going to be your adenosine triphosphate. It's made up of adenine, ribose, and 3 phosphates. This is the principal short-term recyclable energy supply for cells. You can think of it being like the energy currency. RNA is ribonucleic acid. This is usually single-stranded. The 5-carbon sugar is ribose. RNA will use uracil instead of thymine. There are 3 major kinds of RNA. mRNA is messenger RNA, tRNA or transfer RNA, and rRNA or ribosomal RNA. All of them are going to play a major role in protein synthesis, which we will cover more of later.